Thank you very much for the invitation and for the opportunity to present our work. I know that what I present is a little bit different from the usual monthly discussion, which I like to attend. So I would like to encourage everybody who has questions during my presentation to interrupt me and ask if something is unclear, because I do appreciate that quite a few of the things that I'm talking about are different from the usual single-cell RNA sequencing methods. So it's a brief motivation. Why do we want to look at proteins? Because as awesome and as useful RNA sequencing data are, they don't tell us the full story. And we know this from individual important outlier genes, such as the tumor suppressor protein p53, which we know is primarily regulated by protein degradation from and RNA is constitutively expressed. And indeed, when we measure protein and RNA, we see that RNA is not very predictive for the protein abundance. And similarly, when we did a more comprehensive systematic analysis of protein abundances across human tissues, we found that only a fraction of the variance in protein abundances is captured by the RNA level. And in particular, how one cell type is different from another, the relative variation of a particular gene is shown here, is less well predicted by RNA abundances as opposed to the overall abundance of a protein product. And then it's not just protein abundance. What we're really interested to go into is the ability to perform single-cell measurements of protein interactions, degradation, various modifications. We'll actually have a lot more to say about this later this year, of measuring modifications, localization, conformational changes, and so on. So what I'll tell you about today are really initial steps in the direction of this long journey of being able to measure all these important functional quantities at the single cell level. I'm also very interested in this idea that has been articulated many times by different colleagues of being able to use variability naturally observed across single cells to infer regulatory interactions and being able to acquire data sets that are quantitative enough and complete enough with all of the molecules participating in the regulation so that we might be able to do this kind of inference of regulatory interactions. So with this very brief introduction, I'll transition a bit into the technology section, starting with very good news for protein analysis. And this is the well-known fact that proteins are much more abundant than messenger RNAs. And as a consequence, this means that even if our sampling efficiency is modest on the order of 0.1, 0.2%, we might sample substantially more copies from a protein than is commonly possible to do with RNA sequencing, and therefore have less counting error in our estimates. And I'll show you later today data supporting that this notion is indeed has come to fruition. I'll give you this 1 minute, 1 slide introduction to a common approach to mass spectrometry analysis, taking into account that many of you may not be familiar with mass spectrometry proteomics. And most quantitative approaches require some form of protein or peptide separation, which is done by liquid chromatography or capillary electrophoresis commonly. So one of our dimensions here in panel A is simply the separation, the time when peptides elute from the chromatographic column. Then we can measure the mass overcharges of all of the eluting peptides with very high resolution, very high accuracy. And peptides appear as these isotopic clusters shown here in the blow up to the right. So detecting the mass of a peptide ion with arbitrarily high accuracy is insufficient to determine the sequence. So to determine the sequence and therefore link to biology, what we need to do is break the peptide into pieces. And the breakage happens at peptide bonds. So by measuring the product ions of this breaking up of the peptide, we are able to determine the peptide sequence. Just giving you a notion of generally how mass spectrometry works. So with this in mind, there have been 2 different classes of single-cell mass spectrometry approaches developed for proteomics. One approach which we have contributed to is the development of multiplexed methods, where the idea is to do the sample preparation in as high throughput fashion as possible. And I'll tell you today about some improvements in that preparation by a graduate student in my group, Andrew Leduc, whom I can actually see here, and that was funded and supported by CZI. But a common instantiation of this preparation is to simply sort the single cells using a fax sorter, very much in the same manner that one would do this with RNA sequencing. Then the proteins are degraded to, or digested to peptides, and the peptides barcoded. The barcoded peptides then can be pulled together and analyzed by mass spectrometry. And this type of approach allows to analyze a few hundred single cells per day per single instrument, so a few thousand over the course of a week or so per instrument, many more if one has more instruments. And the other line of investigation has been based on label-free analysis, where the aim is to isolate a single cell and then subject the single cell to liquid chromatography mass spectrometry analysis. And there have been many versions of this approach. You're going to hear in the next month webinar from Matthias Mann about a particular approach, label-free approach that his lab has developed. And because in this case, you're not barcoding the cells, the throughput is generally lower. If those methods are applied correctly with the required benchmarks, one can obtain fairly quantitative data. And here I'm showing just one benchmark to give you a sense of what is possible to achieve, where we compare relative protein abundances between two different cell types that are either measured in bulk on the x-axis or measured from single cell lysates on the y-axis. And you can see that the relative protein levels that are estimated with standard bulk approaches agree reasonably well with single cell measurements. And these technologies, while being far less popular and widely applied than single cell RNA sequencing, are actually fairly mature. We have published multiple protocols that are available either via preprints or peer-reviewed articles, and there are websites that you can use to access resources so that you can start implementing those methods yourself. And we also take advantage of protocols IO to deposit all of our new protocols there, so you can access our protocols from this link here. Now, I wrote this review article earlier this year to give a bird's eye view of what are some of the challenges of doing single cell protein analysis and what are some of the solutions proposed so far that have been useful. And the challenges will come as no surprise to this community familiar with single cell analysis. They involve sample preparation. How do we deliver efficiently proteins from a single cell or many thousands of single cells to the instrument for a specific analysis? How do I identify robustly and reliably the sequences of all detected peptides? It turns out that detecting peptides and quantifying them is easier than determining their sequences. How can we scale this up to the entire proteome, to hundreds of thousands, to millions of single cells? And one vignette that I'm going to tell you a bit about is this first challenge of delivering the proteins of single cells to the mass spec instruments. So it turned out that this very simple, well-established approach of sorting the cells into a multi-well plate with a sorter has worked well for us with cell lines, but it did not work well enough for us with the primary samples from human testes as part of our CZI project. So we routinely found that sorted cells gave us very low signal, and that was a painful troubleshooting approach. So to overcome this problem, Andrew Leduc developed a new sample preparation method, a very general sample preparation method, starting with those simple three aims. We wanted to be able to process in parallel in terms of sample preparation many thousands of single cells, many hundreds, thousands, the more the better, because if we can do that, then we are going to have not only high throughput, but less batch effects between different sample preparation sets. We wanted to reduce the volumes so that we have to add less chemicals and therefore less contamination. We can potentially reduce losses of the proteins. And we wanted this design to be completely flexible so that it can be used for a lot of different experiments. And the basic idea behind what Andrew did was to do all of the sample prep in droplets on the surface of a fluorocarbon-coated glass slides. These are basically microscopic glass slides. There's nothing fancy about them except that they have fluorocarbon coating on the top to make them repel both hydrophobic and hydrophilic molecules. And then we can arrange droplets using piezo dispensing that is implemented by Cell N1. So Andrew starts by dispensing a droplet, tiny droplet of a few nanoliters of DMSO, where in each one of these droplets is going to serve for the preparation of a single cell. And then he dispenses a single cell. And this dispensing is much gentler compared to FACS sorting. So that's where the advantage for our CZI project came in. So the FACS sorting that previously resulted in mostly getting cell debris was now replaced with this gentler method that allowed us to recover more proteins for protein analysis. And then the proteins are digested with trypsin while everything is happening inside of this instrument in parallel for all of the droplets, all of the single cells. They're labeled, barcoded, and pulled together. So this approach allows us not only to automate the sample for lots of cells in parallel, but also to reduce volumes below what was previously possible. And here is Andrew. So I would like to tell you a bit of a biological story for which we applied the single cell protein methods. It's not our CZI project because the data from our CZI project are not yet at the level where I would like to start presenting them, but I would like to tell you about this one particular example because it can give you a sense of what single-cell proteomics is capable of doing at the moment. And in this particular case, we were interested to explore the heterogeneity that might emerge as isogenic clonal population of monocytes differentiate into macrophages. And of course, macrophages are very, very diverse cell type of the innate immune system. So we were interested to see if heterogeneity in those cells might emerge even in the absence of polarizing cytokines. And as we did this experiment, we also wanted to compare the results from doing single cell protein analysis to doing RNA analysis with a popular platform, 10x Genomics. So we took cells from the same culture of differentiating monocytes, keeping track of each cell, whether that's a monocyte or macrophage, and analyzed them either by mass spectrometry, single cell mass spectrometry, or by 10x Genomics. And the first thing is the very low bar. What happens if we just project the data into their principal component axis? And here with the protein data, we did that without filtering any proteins, using all roughly 3,000 quantified proteins across multiple single cells so that they can be usefully analyzed. And then we can color code each cell based on its identity, knowing whether it's monocyte or macrophage. And we see that the clusters correspond to the cell types, not surprisingly. Again, that's a low bar validation. And similarly, we can color code cells based on marker proteins. And again, we can recover the ability to identify the cell clusters. If we look at the proteins that are more abundant into monocytes and macrophages, again, as part of our validation, we see what we would have expected. We see that monocytes have higher abundance of proteins involved in cell proliferation, while macrophages have more immune-related proteins, adhesion-related proteins, and so on. What was more exciting for us is to look just at the macrophages and to explore any heterogeneity present in that population that you could actually see on the previous slide, even in the PCA plot, that macrophages were more heterogeneous. And in this case, when we analyze the proteins that are more abundant in either end of the spectrum, we found this very strong correlation to previously established proteins markers of either classically activated, sometimes called M1, or alternatively activated M2 macrophages. And this is the pro-anti-inflammatory axis of activation. And for us, this was a novel and unexpected observation in the system that even in the absence of cytokines, the cells would naturally break the symmetry and go into this continuous gradient of polarized states. So let me see. We also compared our data to 10x genomics. And in this case, we did not measure the RNA and the protein molecules in the same cell. So we needed some way to pair the single cells in which the analysis has been done. And at the first pass, we used correlation vector analysis, simply computing the vectors of pairwise correlations for all genes that are quantified at both the RNA and the protein level, and then correlating these correlation vectors to identified clusters. And we found one cluster where, at least qualitatively, genes had similar abundances at the protein and at the RNA level. And then we also found hundreds of genes for which the opposite was true, that the protein levels showed different abundance from the RNA levels. And these genes here were fairly strongly enriched for signaling functions such as RAB, GTPase activity, and other related functions. We tried using methods for joint dataset analysis, originally developed for RNA sequencing datasets for different datasets, but now use them to combine the protein and the RNA dataset. And in this particular case, we used CONOS, developed by the lab of Peter Kharchenko, to do a joint low-dimensional projection. So in this case, we see that the protein that the cells analyzed by mass spectrometry are labeled in blue and in red, correspondingly for macrophages and monocytes, while the cells analyzed by 10x genomics are in gray. So one thing that you can note from this projection is that the centroids of these clusters are similar, but the RNA cluster appears to be more diffused. And this is a low-dimensional projection that doesn't capture all the variants. We also had more 10x more cells analyzed by 10x genomics. So from this picture, we couldn't conclude immediately that the RNA data are more variable, but we did additional analysis looking at pairwise correlations between RNAs and proteins. And we did find that indeed the protein data exhibit less variability, which is probably due to both technical and biological reasons. Technical because we sampled on average 20-fold more copies per protein than from the corresponding RNA, and biological because some of the transcriptional bursty noise is averaged out by the longer lifetimes of proteins. And here we also tested for possible batch effects remaining due to different biological technical batches of sample preparation, digestion, and so on. And we see that they don't account for much of the variance that is left in the data. Those have been successfully managed and controlled. And I want to go back to this notion that I suggested at the beginning, the thing that I'm quite interested in, using the natural variability between single cells to infer regulatory interactions, essentially thinking of each single cell as corresponding to a particular type of perturbation. And as a very, just to formalize this a little bit, what I would like to do when we have large enough high quality data sets is simply to condition joint distributions based on confounders and ask whether those conditioned joint distributions are the products of the conditional marginal distributions or different, which can allow us to ask for dependence, for example, between kinases. Do they interact directly or indirectly without making specific assumptions about parametric models? So it's a very initial first steps in this direction. We ordered the cells analyzed by single cell mass spectrometry and by RNA sequencing based on the common principal components. And here I'm showing examples of a few genes. Here is NCL, which is a highly abundant protein in transcript that is part of cluster 1, showing similar trends at the RNA and the protein level. And you can see that indeed the data look similar and we have a thoroughly strongly correlated joint distribution. And then if we look at the less abundant protein that again shows similar behavior at RNA and transcript level. Again, you can see the correlated joint distribution, but you begin to see some of the discreteness at the RNA level due to having counted a few messenger RNAs, as opposed to the more continuous abundances measured by mass spectrometry of the more abundant protein. Now comes a more interesting protein. If we look at p53, we see that the RNA and the protein levels are not correlated, which to a large extent confirms the expectation that p53 is regulated by degradation rather than the abundance of its transcript. And we wanted to go one step further and ask whether the messenger RNA of p53 is informative for the transcriptional activity of the protein, or the protein level will be more informative. And to ask this question, we simply correlated the messenger RNA to well-annotated targets of the transcription factor that are either activated or repressed. And we essentially saw no signal from this analysis. We saw that we couldn't predict the abundance of targets of p53 based on the messenger RNA for the transcription factor. And when we did similar analysis with the protein, we found that we did have some signal for the repressed targets, that they were significantly negatively correlated to the protein abundance, and we did not see signal for the positive targets. And in this case, it's easy to hypothesize of possible causes for that, which include the alignment of the data sets, include post-translational modifications of p53, which we did not measure, localization, additional transcription factors that regulate those genes, and lots of other related factors. But I found this observation nonetheless encouraging, that we are able to measure more information about the regulatory activity of the transcription factor by measuring the protein abundance compared to the transcript level. So at the very end, I want to return a bit to the technological aspect. Having told you what we can do now, I want to tell you that this is nowhere close to the asymptote of the field. This is not mature. These are just early baby steps. And for that, I'll just introduce to you how a peptide looks to the mass spec instrument. It looks like this peptide feature. So this is a data object composed of usually hundreds of measurements. It's not a blip on the detector. Essentially, we measure all of the ions that do not have heavy isotopes. And then the second isotopic peak corresponds to having only one heavy isotope in the molecule, having three, and so on. And this is measured across lots of survey scans. This is how a peptide-like ion looks like. And the reason why I'm telling you this technical detail is the fact that we can see over 60,000 peptide-like ions in a single cell injected for mass spec analysis. We don't yet identify the sequences of all of them. We don't yet identify all of their modifications, but I can think of a lot of ways to do that, and that's something that is going to happen, and that's why I wanted to tell you to watch out for it, to sort of tell you that what I presented at the moment is representative for the current state of the technology, which is becoming useful for biological investigations, but it's no way reflective of some asymptote that we are reaching in terms of having used up the limited sensitivity of the mass spec available for us. Everything I told you about was product of a very fun collaboration of these people here that make up my group at Northeastern. And they'll finish by acknowledging the important role that funders, visionary funders have played in supporting our work, particularly the NIH Directors Award, the Allen Distinguished Investigator Award, and of course the Chan Zuckerberg Initiative support for our work, which was very useful for coming up with this next generation sample prep that I described earlier today. So I'll stop here and take any questions that you may have.
Nikolai, I have a question for you. So how confident are you in the ability to measure low abundant proteins?
What we have measured so far has been very much prioritized based on protein abundance. in the sense that we instruct the instrument, when you see more ions than you have time for to analyze, how are you going to choose? We tell the instrument, use a heuristic, pick the most abundant first, the second most abundant second, the third most abundant third, and so on. And we don't have to do that, but we've done it because in the absence of other information, that's a convenient heuristic. And that's one of the reasons to sway our measurements in favor of more abundant proteins. Now, we've used another heuristic, not yet published, but we're going to talk about it at the Single Cell Proteomics conference about a month from now. Actually, Gray Huffman, a graduate student in my group, has used it, where instead of using this heuristic of picking the most abundant peptides, we can tell the instrument, we have this list of 5,000 peptides of interest. We want to prioritize their analysis rather than just the most abundant peptides. And that's one way to even now at the moment, to measure less abundant proteins of interest. Now, as you start doing that, of course, you get to the limit of what if you don't have signal? What if mass spec is not sensitive enough? What's really that limit at the moment? And depending on how you do the data acquisition and this and that, at the moment, I think we have about 0.1% efficiency, maybe up to 1% efficiency of delivering peptides to the instrument. So that means that the protein needs to have at least 1,000 to 10,000 copies to be analyzed reliably. And most of the losses at the moment occur from ionization being relatively inefficient process. But there have been published prototypes of increasing the efficiency from 1% to 50% or so. And it's just, I think it's just a matter of time until those are being incorporated into widely used commercial instruments. At the moment, these are prototypes used in labs specializing in the tech development. So at the moment, the simple answer is, if your protein of interest is present at level of a few thousand copies per cell, we have a good chance of quantifying it without a promise. The higher abundance, the higher the probability of quantifying it. Of course, it becomes easy.
I have a quick question, if I may. This is Chris Sander. Impressive progress. I just wonder, in terms of phosphopeptides, what's the current limit in terms of the number of cells that would be needed? I imagine not single cell to see phosphopeptides with some reliability.
So it is actually a single cell, the sensitivity. The question becomes how many phosphopeptides. In single cell, so if you just take the data that I presented today and do a variable search for phosphopeptide, which is standard analysis practice, we can see a few hundred phosphopeptides without having done anything special. In our data, we already see that. And a few hundreds, they're seen across many cells, so there would be a lot of missing data. But if one comes up with a better method, data acquisition, which stay tuned, it's coming, then across single cell, in individual single cells, we can quantify hundreds and even thousands of phosphopeptides. And then the question becomes, what if you want deep coverage? Then potentially you may want to have larger number of cells to analyze, and then the exact number of cells becomes contingent on what is the sensitivity that you want to have, how many phosphopeptides. But even as of now, we do have clearly the sensitivity in some of our published data to quantify phosphopeptides.